Hello and welcome to my channel and my first video, which will be an introductory video on immunology. Do you want to know what historical event marked the beginning of immunology? Why sometimes it takes longer to get better after infection and sometimes this happens more rapidly? Is smallpox extinct? If you want to find the answers, watch till the end. Please like the video, comment, subscribe and ring the bell to get notified if you want to watch similar videos in the future. Without further ado, in this brief video I'll introduce you to some of the basic principles, concepts and notions in the field of immunology. You will learn about the levels of defense which our immune system possesses and the cells and molecules it utilizes in its warfare against infection. Okay, so first of all, let's begin with the definition. What is immunology? Immunology is the study of the body's defense mechanisms against infection. So, if we approach the understanding of immunology chronologically, we will begin with ancient Greece. Even back then existed the notion of immunity. They observed that surviving a disease provided better protection against the same infection in the future. The most prominent figure in the field of immunology, the father of immunology and vaccination if you may, is Edward Jenner. What he did was that in 1796 he proved that inoculation with cowpox protected the recipient from smallpox. He performed this risky intervention on his healthy 8 years old gardener's son. Well, what can we say? He was the father of immunology, not ethics. But at the end of the day, smallpox was eradicated entirely after the introduction of the vaccine. Or is it? The answer you'll find by the end of the video. Moving forward, the practice of vaccination continued evolving, and later, Louis Pasteur, I'm not sure if I'm pronouncing this correctly, so excuse my French. So, Louis Pasteur invented the vaccine against rabies. Okay, so before I completely bore you with history, I will mention one last discovery and we will move on to the more intriguing part. So, Emil von Behring and Shibasaburo Kitasato, oh gosh, I don't know if I pronounced this correctly too. Anyways, they discovered that the serum of animals immune to diphtheria and tetanus contained a specific antitoxic activity that provided short-lasting protection against those diseases in humans. The constituents of the serum responsible for this antitoxic activity are antibodies. They bind to specific toxins and neutralize them. Now, let us look at the barriers that pathogens need to overthrow to achieve their goal. Use our resources for their growth and proliferation. But our immune system will not allow this. It employs several lines of defense. So, first line, or first barrier, anatomical barriers, skin, oral mucosa, respiratory epithelium, intestines. They act more like a passive defense because they protect us from exposure to pathogens. However, mucosa synthesizes some antimicrobial proteins which function like natural antibiotics. This level of immune response can be fatal to many pathogens. However, if they manage to breach or evade it, they will encounter the second barrier. Now we are looking at the second barrier. The second barrier comprises antimicrobial proteins and the complement response. As I already mentioned, the antimicrobial proteins function like natural antibiotics. But the complement comprises 30 plasma proteins and it can join forces with antibodies to lyse bacteria. In the absence of antibodies, complement proteins contribute to the innate immune response. So, what is the innate immune system? I'm glad you asked. This is the third barrier or the second line of defense. The main warriors at this stage are cells, the white blood cells, leukocytes, and signaling molecules. Everything begins with interaction between the pathogen and the sensor cell. Imagine it that way. There is a foreign agent in the organism, the pathogen, and for it to be disposed, it needs to be recognized by sensor cells. How this happens, what chain of events this triggers, we'll discuss in the next slide. Okay, as I mentioned, there needs to be sensor cells and pathogen, or rather the other way around. There needs to be an invading pathogen and the sensor cell needs to recognize it. How do sensor cells achieve that? How do they recognize the pathogen? Well, it's thanks to their PRRs. Don't get scared, this abbreviation means pattern recognition receptors. Those receptors are limited and invariant. They're characteristic for the innate immune system. They recognize simple molecules and regular patterns of molecule structure, or PAMPs. This abbreviation means pathogen-associated molecular patterns. The host cells do not have such structures, remember that. They are present only on pathogens, therefore allowing the correct discrimination between self and non-self. Remember, this is a very important property of the immune system, correctly distinguishing between self and non-self. Example of PAMPs include mannose, a peptidoglycan. Once the PRRs on sensor cells recognize PAMs, they become activated and innate immune response is initiated. Sensor cells can undertake two possible paths: phagocytose, or simply said, engulf the pathogen and digest it, or the other option, synthesize inflammatory mediators, chemokines and cytokines, which convey signals to other immune cells. As a result, more leukocytes are recruited into the site of infection to eliminate the pathogen or tissue damage. On this slide, you can see a diagram illustrating hematopoiesis. You can stop the video here and peruse the origin of the differentiated cells of the immune system, but I'll just mention that B lymphocytes mature in the bone marrow, while T lymphocytes mature in the thymus. Now let's observe the cells that eliminate pathogens. Let's begin with phagocytes. They phagocytose, aka engulf and digest foreign agents and infected cells. There are three types of phagocytes: macrophages, granulocytes, and dendritic cells. Macrophages reside in almost all tissues, and they have specific names in some sites, like for example they're called microglia in nervous tissue. They arise during embryonic development, but can also arise from monocytes during infection. They also recognize pathogens coated with complement proteins and phagocytose them. And so do neutrophils. The most important function of dendritic cells is presenting antigens. Remember that. Their most important function is presenting antigens. They also synthesize inflammatory mediators, but they can also carry phagocytosis themselves. They initiate adaptive immune response. Neutrophils, eosinophils, and basophils are collectively termed granulocytes. Neutrophils are easily recognizable because of the distinctive shape of their nucleus. The largest proportion of leukocytes in the blood are neutrophils. Eosinophils kill parasites coated with antibodies. Negative sides of eosinophils and basophils is that they can participate in the occurrence of allergic reactions. Natural killer cells are crucial in the initial innate response to viral infections. Now it's time to look at the third line of defense. Adaptive immunity is the major evolutionary advantage of vertebrates. When the foreign agent is introduced to the adaptive immune system for the first time, the response is considerably slower. However, upon a subsequent exposure to the agent in question, the adaptive immunity mobilizes significantly faster than the first time. We have immunological memory owing to the adaptive immune system. What differentiates the adaptive immunity from the innate is the antigen-specific receptors of adaptive immune cells and the products of plasma cells, antibodies. Lymphocytes are categorized depending on whether or not they have encountered an antigen, complementary to their antigen receptors. If they haven't, they are referred to as naive. If they have, they are called effector lymphocytes. Naive T cells can differentiate into three types of activated or effector cells: cytotoxic T lymphocytes, regulatory, and helper T lymphocytes. B naive lymphocytes differentiate into plasma cells and secrete antibodies. When we talk about antibodies, it's important to mention their protein structure. They constitute variable and constant regions, but this is beyond the scope of this video. To fortify, activated B cells secrete antibodies, or effector B cells. Those are synonymous. Those antibodies can aid the elimination of pathogens in three ways: neutralization, opsonization, and complement activation. During the maturation of lymphocytes, it's a matter of life and self-destructive disease for lymphocytes with self-reactive receptors to be destroyed. It's important, they need to be. If they're not destroyed, they can target the host's own tissues, leading to the development of autoimmune diseases. We saw various ways in which our immune cells can recognize extracellular invaders. Detecting the invader is crucial in eliminating it, and the task of finding it within cells complicates the whole process. So how does our immune system find those intracellular invaders? Through extracellular representation. This all happens thanks to the major histocompatibility complex. It encodes glycoproteins responsible for presenting peptide antigen so that it can be recognized by T cells. The MHC receptors are situated on the membrane of sensor cells. T cells have two types of receptors, CD8+ and CD4+. Now it's time to close this video with the question I left unanswered in the beginning. Did we get rid of smallpox for good? Well, yes and no. There are no active cases, and the last one was more than 40 years ago. However, samples of the pathogen causing smallpox, the variola virus, are preserved in laboratories. What does this entail for the population? Well, let's hope there are no clumsy people or such with destructive urges working around those samples, because if there are, the humanity will encounter a disaster with immeasurable consequences, because it has been decades since people were last vaccinated against the disease. On a positive note, the fact that with improved healthcare and awareness of science, we have achieved the disappearance of one of the most fatal diseases to ever exist. This serves as a proof that further innovations can facilitate the ultimate victory against many other diseases still existing today. Thank you for watching, and I hope you found this interesting. If you liked it, please put thumbs up, comment what topic you would like to learn more about next time, and subscribe. Oh, and click the notification button. Hope to see you soon.